Welcome to Behavior and Molecular Assessment of Sleep-like States in Mosquitoes. Mosquitoes are efficient vectors at transmitting harmful diseases such as malaria and yellow fever. More than half the world's population is at risk of mosquito-borne diseases. Therefore, it is important to study mosquito behavior and ecology to further enhance and improve existing disease models and control strategies. In fruit flies, sleep deprivation studies revealed increased immunity response to diseases, so stronger immune system, and also decreased aggression. However, sleep has largely been understudied in mosquitoes. Furthermore, we know that the circadian rhythm largely influences resting and active cycles. Circadian rhythm also influences biting frequency or feeding behavior and immunity, as well as other aspects of mosquito biology. More importantly, sleep may impact the ability of mosquitoes to transmit diseases due to factors like biting frequency and immunity, similar to that of fruit flies sleep studies. To characterize that sleep-like state does occur in mosquitoes, we examine postural difference, rest and active profile, rest rebound, and ortholog analysis. Figure A reveals the difference in posture of active and resting states in Aedes aegypti. We can see that the angle between the main body and the orientation of the hind legs show the difference between these postures. According to Figure B analysts, there is a unique difference in posture between the active and the sleep-like states in Aedes aegypti. In Figure A, we can see that the Aedes aegypti rapidly increase in activity starting midday and peak right before dark transition. The activity very quickly declines after the peak, and low activity occurs throughout the night. Similarly, in Figure B, the Culex's activity peaks right before dark transition. However, Culex's remain more active during the night and less active during the morning when comparing to Aedes. On the other hand, Figure C reveals that Anopheles stephenses are very active during the night, peaking in the middle of the night, but are very non-active during midday and evening. These laboratory measurements of rest and active cycles match the field observations of each mosquito species. Figure A is a basic profile of sleep deprivations of Aedes aegypti. We can see that the activity and resting cycles are normal in the first 36 hours. However, mechanical vibrations during the 36 to 48 hours occurred, which cause abnormally high activity when normally rested occurs. In Figure B, the box plot compares the before and after sleep deprivation, which results in a significant p-value. This reveals that the Aedes aegypti were less active after sleep deprivation. 58 Drosophila sleep-linked genes have orthologs in 5 mosquito species. Reanalysis of the previous expression data reveal putative sleep-associated genes in mosquitoes. Ultimately, these studies suggest that a sleep-like state does occur in mosquitoes, and further understanding will help improve existing models and control strategies. Our future direction is to understand the genetic basis of mosquito sleep and the effect of sleep deprivation on mosquito physiology, and also the assessing of effect of sleep deprivation in mosquitoes on vectorial capacity and disease transmission.